Good morning. Thank you for being here. I'm very pleased this morning that you all got here with the snow and so forth, but to introduce our speaker, Dr. Jeffrey Drazen. Dr. Drazen, as I think most of you know, is the editor-in-chief for the New England Journal of Medicine. During his tenure, the journal's published major papers advancing the science of medicine, including the first description of SARS, timely coverage of the Ebola epidemic, and modifications in the treatment of cancer, cardiovascular disease, and lung disease. It's been at the forefront and has the highest impact factor of any journal publishing original research. Well, Dr. Drazen is not only the editor-in-chief of New England Journal, he was also an active researcher in pulmonary medicine and defined the role of novel endogenous chemical agents in asthma. This led to 4 new pharmaceuticals for asthma, which are now used in the treatment of millions of patients. By way of background, Dr. Drazen graduated from Harvard Medical School in 1972. He's from Missouri. He served his medical internship and residency at the Peter Bent Brigham in Boston. That's what it was called then, the Peter Bent Brigham. There were 2 Brigham hospitals, actually. And he was a clinical fellow and a research fellow at Harvard Medical School and the Harvard School of Public Health. He served as chief of pulmonary medicine at the Beth Israel, and then chief of the combined pulmonary divisions at the Beth Israel and Brigham, and then as chief of pulmonary medicine at Brigham and Women's Hospital. So I'm very pleased today to have Dr. Drazen. He may have a surprise seminar for us. Mm-hmm. Okay.
Right.
Welcome.
Right. Thank you, Teddy. In my youth, I learned that if I got to synagogue early, I could get a good seat in the back. And then later on in my thespian days, I learned that if I got to the theater early, I could get a good seat in the front. So this looks like it's going to be more of a religious than a theatrical experience this morning. But we're going to vote. So everybody needs to wake up. All right. I'm going to give you 3 choices about what you're going to hear about for the next hour. They're very different sorts of things. And at the end of this time, I'm going to ask you to vote. And everybody has to vote. You can't abstain. Okay. So you're going to have to make up your mind. Choice 1, and these are in chronological order of the way they were prepared, is a talk about how we go from what we think to what we know. I call it the rise of medical evidence. We use tuberculosis as an example that walks us through this. So that's choice 1. Everybody got that? The rise of medical evidence using tuberculosis. Choice 2 was that the year 2014 was the 50th anniversary of what major public health report published in the U.S.? The Surgeon General's report on smoking and health. And so I prepared a lecture about how epidemiological evidence has been gathered and used to shape clinical thinking, because we don't do clinical trials in humans where we assign people to smoking and non-smoking groups and follow them for 50 years. So we reviewed that evidence. All right, so that's the what I call the role of medical information in the war against smoking. And the third is about the West African Ebola outbreak and what we know about this virus historically. History is fairly short since it was identified in 1976, and a synthesis of kind of where we are now with this. So that's kind of more current events. So has everybody got all 3 choices? All right, so you can't, even if you come in late, you have to vote. Even if this is just like, just like it is in our national elections. So what we're going to do is ask for a show of hands for those who want to hear about the rise of medical evidence. Let's see here. Okay, we're going to sort of see. Next is who wants to hear about smoking. All right, we have very few smoking. And Ebola. And we're sort of tied. I think what I'll do is I'll talk about the rise of medical evidence, and then if people really want to hear, we talk a little bit about Ebola at the end, depending on how we do. So let me mess with the electronics here. let's see here. So imagine that you're sick, you're not feeling too well, you go to see the doctor, you tell her about these complaints, and she says, I'm not quite sure what's wrong with you, you have me puzzled, but if you come into my office, in the back, I have a secret machine that I can use to make a diagnosis. It's really, really reliable, and I can use that same machine to determine your treatment. You're feeling pretty bad. You've been to a couple of doctors. What are you going to say? Okay, let's do it, right? So you go in, and this is the machine. How would you feel? Would you leave and want to have a spin for your treatment after we figure out what's wrong with you? Well, what I want to let you think about is that much of what we do in medicine has been the play of chance. We think we're not being governed by the play of chance, but we, in fact, are without knowing it. And how do we really know what we know? How do we get the evidence that drives our thinking so we can go from something that we think we know to something that we know we know? And I started off life as a physicist, and people would develop a theory, but then they would test it. And if the test didn't work, they'd throw it out and build a new theory. We don't do that in biology because the tests usually aren't all that precise to tell the theories apart. We're getting better at it, but that's the story. So medicine's taught by example. So let's talk about an example. Imagine it's 1812, the year the New England Journal was formed, and you live in the city of New York. You're a young man, and your cousin is running a business in Boston, and back then, and it's actually true today, it had to do with handling money, and he wants his family, he can trust his family, he thinks, to come. So he invites you to go from New York to Boston for this great new job. You're not terribly well off. If you were terribly well off, you'd take the boat. But you're not terribly well off, so you take the stagecoach, and for 6 days, you're cooped up with the same 8 people. And just your luck, the guy sitting next to you, like that for 6 days. That's just your bad luck. And there you are. This is an image I picked, shown. It's an oil by Anne-Louise Giraudoux de Rosy-Trielson, painted about that time. This guy doesn't look so good, like he's a little sweaty. He goes to see the doc. He says, I'm telling you about the stagecoach ride. I've never felt good after that. I've been having a cough. I'm bringing up phlegm. Sometimes there's blood in it. He probably didn't use the term hemoptysis. I'm just sweating at night. I've been losing weight. I feel like dirt. So the doctor does an examination, and with the tools available to him at that time, and most doctors were he's at that time, there's loss of muscle mass in the upper pectoral region. The doctor makes a diagnosis. What's the diagnosis? Phthisis, right? Pulmonary consumption. Tuberculosis, as we know it now. That's the doctor's diagnosis. But the doctor is just playing the odds, right? This is the abstract of the bill of mortality for the town of Boston from the 22nd of October, 1810 to the 31st of December, 1811. Period of slightly over a year. And if you look, you see the most common cause of death was consumption. 221 deaths out of a total of 942. Nearly a quarter of the people who died from tuberculosis. So the doc knows the symptoms of tuberculosis. He's heard your story. It's highly likely that he's right. But it's a guess. Right? This diagnosis has not been confirmed. It's a guess. And these are some of the treatments that were taken from the New England Journal of Medicine. You could breathe the vapors of boiling tar. And if you lived through that, you probably would live through your pulmonary consumption. You can take preparations of gold. We'll see they were actually used until quite recently. Preparations of silver. Or you could go to take the sea. And this is a letter about a physician who had sent his patient to Flanders to a floating infirmary for a year of rest. and every day they'd be rowed out and would do nothing but wait for their TB to be cured. Now, tuberculosis is a great example for this sort of thing, because if you take people who are otherwise healthy and they get tuberculosis, about half of them get clinically better without us doing anything. Now, we now have ways of determining they have latent tuberculosis, but back in 1800, they looked better. Maybe a little sickly, but they'd been sick for a year. So there were all these kinds of treatments out there. So then what happened between 1812 and 1912? We learned something. Right? In that century, we learned a lot. And people like you, people who were students trying to understand things, began to understand this disease, and knowledge mitigated the play of chance in the diagnosis of tuberculosis. And so let's just spend some time and look at the knowledge that was gathered. There are seats up front. If you'd rather stand in the back so you can sneak out, it's okay. But if you want to sit down, that's fine too. So it all started with Pasteur. Actually, it should have started a little before Pasteur with Ignaz Semmelweis, the Hungarian obstetrician. You guys know that story? Right? Who knows the Semmelweis story? I'll tell it for the people who don't know. So he's an obstetrician. There were two major obstetrical hospitals, one in Buda and one in Pest, and I'm pretty sure he was in Pest. And his patients were the poor patients of the city. But they had very few deaths from childbed fever, the mother dying after birth of infection, which we now know is most commonly streptococcal infection. And Semmelweis had his doctors washing their hands before and after and during the procedures. They were trying to keep their hands clean. And their women did much better than the women at the fancier hospitals where the doctors were all haughty and they didn't do that. Now, Semmelweis was laughed out of business. He ended up in an insane asylum. And the reason was that there was no theory to explain why washing your hands should make a difference. You know, it seemed like voodoo. You know, getting the Ebola story. Why did Ebola spread in West Africa? Think about it. You know, people suddenly start to die. It's the devil working. We don't understand it. So when Semmelweis came around, he didn't get anywhere because he didn't have a theory that made sense, that had been proven scientifically. It was Pasteur who elucidates the germ theory of disease in the early 1860s. Right? How many people have been to the Institut Pasteur? It's there on the Rue de Docteur Roux in Paris. It's a great trip for those of you who go. His famous experiment of the swan's neck flask, where he took 3 flasks. He took beef and he boiled it to make beef broth. He left one open to the environment, and a day later it was putrid. The second one he sealed, and it stays sterile to this day. In the third, he attached a long glass tube, very thin, about 1.5 millimeters in internal diameter, a couple of maybe 40, 50 centimeters long, a swan's neck, as he called it, and that stayed sterile. In fact, that experiment is still ongoing, and if you go to the Institut Pasteur, you can see it. It's in the humidified case, so the liquid doesn't evaporate. And that stayed sterile. So from that, he inferred that there were germs, things that we couldn't see, but that were in the air that caused putrefaction. And with this idea that there were invisible things that could cause it, he established the germ theory of disease. So then we get to this man, Jean-Antoine Villemont. Now, he is a French military surgeon, a student of Pasteur's, and he does an experiment. Remember, tuberculosis had been around for quite a long time. We saw the people dying from it in Boston. That was early 1800s. It's now the mid-1800s. And he takes a person who's died from tuberculosis at autopsy, the cheesy material from their lung, and puts it in a rabbit. And the rabbit develops tuberculosis. And from this, he concludes la tuberculose est contagieuse, donc évitable. The tuberculosis is contagious and therefore preventable. And that was a step forward, because before that time, people had no idea. They knew that when you were in high density populations, developed it. But with Pasteur and then Villemont, they really understood this idea. Now, the other thing that's sort of interesting about this is that for centuries, people have been raising money for tuberculosis through selling stamps. And this stamp here is Achetez le nouveau timbre anti-tuberculose. Buy the new anti-tuberculosis stamp. It was 10 francs. And a little money went to the sanitarium. Some went to the post office. But they were raising money for this thing. This is Villemont. But it took, that was 1865 or so. It took 1882. Robert Koch, working in Berlin, identifies the tubercle bacillus as the cause of tuberculosis. But he goes beyond that because he becomes the father of causality. Right, what we're all hoping is that we identify something here and we say it's the cause of this. And when we do a lot of epidemiology, I give Kay a hard time about this all the time, because epidemiology can almost never establish causality. In the smoking talk, which you decided not to hear about, was mostly about how we got over that big break. In the smoking, the tobacco companies used that scientific fact that you couldn't establish causality with epidemiology to sell cigarettes and sell death for quite a long time before people finally could be convinced by that. But so Koch identifies this and he sets up his postulates to show that an agent causes a disease. It has to be recoverable, it has to be transmissible, you have to be able to give it to a fresh animal and get the disease again. And this are his drawings of the tuberculous bacillus. He's working through a optical microscope at the limits of resolution available in the 1880s. And this is a scanning EM of the tubercle bacillus taken sometime in the year 2000. And I think he's got a pretty good job of identifying the tubercle bacillus. Now, interesting, he gets the Nobel Prize in 1905. He announces his discovery in 1882, and 23 years later, he gets the Nobel Prize. Now, these were two of his students. Right? We had Villemont, was a student of Pasteur. We have Zeil and Nielsen, who were students of Koch. And they learned that the method used to isolate and to identify the tubercle bacillus by Koch was so complicated that it would never be used clinically, because it was just something that you weren't going to be able to do. Now, back then, clinical labs were much simpler. Doctors did them themselves. And show how long ago this was. When I was an intern in 1972, we did our own AFB stains. Now you can't get near a lab to do it, but these guys developed a stain. It's called the Zeil-Nielsen stain, and here in the year 2015, it still represents what many people consider, we're now in an age of transition, the gold standard for the diagnosis of tuberculosis. This is a smear stained with this material, and it shows up the organism as red on a blue background, and you wash it with acid, but the stain doesn't wash out of the tubercle bacillus, and that's why it's called the acid-fast stain, and why we used to refer to these items as in my training as a red snapper. Not a fish, but this method is still being used today. So these guys worked to do research that was replicable and applicable. Now, we hear a lot now about the replicability of research, about how bad it is. And I think the difference between 1880 and 2010 is that people are trying to get at the truth, and they know that other people want to use that information to advance the art. When I was a student, I remember coming down here, Saul Permut was I think in this school, and we would have lots of arguments and talk, but the goal was to make sure that what you did was right, because nothing was more shameful than to stand up and say A, B, C, and have it turn out to be wrong. You wanted it to be right, and so that's what we need to do. These guys wanted it to be right. It has been right, and we've been using it ever since. But somebody had to make sputum. You had to do a smear to see it. It took this guy. You probably know. This is Rankin, Right? He's a physicist, not a physician, and he's working in his lab with this thing. It's a Crookes tube, and this is the cathode here, and this is the anode, and when you fire this up, the electrons go zooming this way, and they can't really all turn the corner, and so some of them come out the front. And this is what physicists were using in the 1890s, and they were using it actually to probe the structure of the atom. But he realized that if he interposed his hand between this ray source and a phosphorescent screen, which people were using to look at these diffraction patterns, he could see his bones. So this was something different from what all the other physicists were doing with these rays. And so he calls his boss and he says, look, this is important. I want to get the year off to study them. And his boss was a smart guy, gave it to him. So this is the first X-ray picture ever taken. I can just imagine his wife was his assistant. And so I can say, hold your hand still, honey, I want to take a picture. Right? So she takes this picture, and this is her wedding ring here. And this is the first X-ray ever taken. Now, I've been told, I wasn't around at the time, that after she saw this, she said, ugh, I'm not working with you anymore. I don't want to see my bones. And she was out of the lab. They found somebody else to help them. So I gave my wife a New England Journal calendar, and she looked at it and said, I don't want that thing in my house. It's all those disgusting pictures. So with this, we're able to see inside the body. And he gets the Nobel Prize. This first X-ray was published in 1895. 6 years later, he gets the Nobel Prize. Right? Took Koch 23 years. Took him 6. Easier to replicate. It's physics, not biology. Okay. So now the diagnosis of tuberculosis can be more certain, and TB becomes the focus of public health. So this is they kept really good records in England and Wales in terms of the cause of death. So we have information from the 1840s to the 1960s with a reasonably similar case definition. And you looked at the death rates, and they'd fallen by a lot, even without understanding, by getting rid of crowding. But we have Villemont here in 1860s, and now we understand that it's a contagious disease. And these rates just fall to World War II, to World War I, and then they go through a pause. They pick up again. We don't introduce treatment for tuberculosis until 1948. Effective therapy for tuberculosis. What do we do for people with tuberculosis? You figure out who they are, and get them out of town. That was the way to treat them. Now, how did you do that? Because it wasn't cheap. Because they'd take somebody who was, and public health was allowed to do this. It's a disease where you can remove somebody from society, and they did it by having a crusade. So this is the international symbol for tuberculosis, and it's the cross that was used during the Crusades. And the Danes started off with this idea of selling stamps. Remember, I showed you the Villemont stamp for tuberculosis. This is a Danish stamp, and this is the right time of year. They used them at Christmas time, and they started off by selling you a stamp which wasn't a postage stamp, but you put it on a holiday greeting to show that you cared. The Cross of Lorraine. And the American Red Cross, and later the American Lung Association. In fact, they still apparently sell Christmas seals, and I looked at all my Christmas cards this year who come from doctors, many of whom are lung doctors. Nobody had a Christmas seal on it. But in my family, even though we were Jewish, we would send out cards at this time of year with a Christmas seal stamp on them because it was considered a way of showing your support for the community. They were raising money for TB sanitaria and for these anti-tuberculosis campaigns, and you can see that they had all sorts of things like this. They were really trying to get the message out that this is bad stuff and you have to do something to prevent it. You have the ghost trying to enter here, you have the nurse, you have the, I'm not quite sure, the breadwinner here keeping the door closed. But this is it, fans. This is why you're here. Tuberculosis robs you and public health protects you. Right? And you're here to protect populations. That's what you learn to do. So this is what that money went to do. They built sanitaria. Some of them are private sanitaria, some of them are public. This is a sanitarium of Dr. Turbin in Davos. This is another one in Hungary. This is one in northern China, but you see it's got the, it's a TB sanitarium right there in the front door. I didn't get a picture of it, but I was in Havana at the eye hospital. The Cubans are really big. They do almost all the eye care in the Caribbean basin for people that are can get into Cuba, which I guess we can go now ourselves. And you walk into this building, which is adjacent to the hospital, which is a big hotel for the people who are having eye surgery in their family, and there on the floor is the Cross of Lorraine. So I said to my guide, "Oh, this must be an old TB hospital." And she said, "Yes, how did you know?" I said, "Look at the floor." And they aren't that big into redoing infrastructure. But here is a tuberculosis sanitarium in Saskatchewan, in Texas, in Syracuse, New York. And they're all pretty much the same. They face, in the northern hemisphere, they face the south. In the southern hemisphere, they face the north. They face the sun. People had figured out that the sun was good for you. We now think it probably is related to vitamin D. Many people were, in fact, vitamin D deficient, and fresh air. And so even during the winter, the windows are open, and this is a ward, typical tuberculosis ward, woman's ward, men's ward. This is a particularly interesting one, is that local businesses funded these beds, and that's what the sign up here was for. It's just like it is now. But these are all these patients, and they're all out of town, which is what people wanted because they had tuberculosis. They weren't spreading it in the community. This is National Jewish Hospital in Denver. Now, going into delicatessens, when I was a kid, there was always a little metal box that you could put your spare change in, and it went to build this place, which offered free care for patients with tuberculosis. It's still one of the tuberculosis centers, referral centers for the U.S. They don't have wards like this anymore, but this is what it was, outside facing south. And so now let's imagine it's a century later, right? It's 1900s. We have our same story. You got the guy in New York, his uncle calls him and says, "I got a great job for you in Boston." This time you take the train, right? Instead of taking days, it's going to take maybe a day. And instead of being crammed in a car with 6 people, you're crammed in a car with 60 people. But just your luck, the guy sitting next to you, like that, for the entire trip. So you get to Boston, and now I have a contemporary drawing from John Singer Sargent around 1900. This guy doesn't look so good. Right? It's the same story. He had that train ride, and he's had fever because we now have clinical thermometry, right? That was introduced in the mid-1800s. Thermometers. I'll stand back and tell a story. How many of you have read The Magic Mountain, Der Zauberberg? Right? It's a great book by Thomas Mann. Anytime you have a spare 3 months, you should read this book. It's full of words, but it's right, nothing else to do, right? He won the Nobel Prize for Literature, but it's a story of this guy, Hans Castorp, and how he goes to visit his friend, his cousin actually, and turns out that it was in a TB sanitarium in Davos, Switzerland. And Davos, Switzerland was the highest inhabited city in Europe, and people had figured out by that time that height was good because low PO2 made it hard for these organisms to grow, so the higher you were, the lower the PO2. And it turns out that when he's there, he had tuberculosis. He probably had it when he came, he didn't get it there, but that he has his temperature taken. And first he tells the story of buying a thermometer. Every patient had their own thermometer, and how he was a rich guy, so he paid a very expensive thermometer and had his temperature taken 5 times a day. Because doctors had determined, it's still true as you'll see, that body temperature was a harbinger of people who did poorly. If your body temperature was elevated, things didn't look good for you. So something you could measure, so now we can measure fever. We didn't have it before. Cough and sputum production, hemoptysis, night sweats, weight loss, same stuff our patient had a century before that. But now the doctor, in addition to looking at loss of muscle mass, can hear abnormalities in the lung, right? Because we had René Laennec, invented the stethoscope, 1819. D'auscultation immédiate, replaced by d'auscultation replaced d'auscultation immédiate, with the ear to the patient. And so you can hear these abnormal sounds. You have the chest x-ray. You can do a sputum culture. And in this case, here's our case here. This is a tuberculosis x-ray. You'll be able to turn off the lights, but if that's not easy to do, we'll just ignore it. So this is tuberculosis x-ray published in the New England Journal of Medicine in 1910. And you can see these are these fibroproductive infiltrates that are characteristic of TB. And those of you who are medically trained are looking at this film and saying, this is really strange. And that's because it looks like his heart's in the wrong place. But this was an x-ray that was published in 1910 before we'd established a convention to show the left side of the patient on the right side of the film. So this is not a case of situs inversus. This is what it was. And so it illustrates the point of how important it is to have conventions that everybody agrees on when you're sharing information. Because you're looking at this information and you're saying, gee, this guy's heart's in the wrong place. But no, the x-ray is just flipped around because we hadn't agreed yet about how to communicate information. Now, if I could just get the medical record vendors to agree to exchange information, when I'm working in Boston and somebody sees me in consultation from some other city, I could get their medical record information without having to carry it. But here in 2015, we still don't have interoperability because we don't have conventions. Conventions are really important. So that's what we see here. And you have now a positive sputum spear Ziehl-Neelsen stain. This guy has tuberculosis. You don't think he has tuberculosis. You know he has tuberculosis. And that's a big step forward, going from something you think to something you know. So then the cynic says, so what are you going to do about it? Right? You made a diagnosis. I came for treatment. Right? So the diagnosis is based on firm knowledge. And public health decreases TB spread, but therapy still left to play a chance. So this is the New England Journal of Medicine's forerunner, the Boston Medical and Surgical Journal. This is just a brief aside. In 1812, we were founded by John Collins Warren. Came back from Europe, realized Boston was a medical backwater, formed a club where every Thursday they talked about medical information. After a couple of years, they decided to start a magazine. They called it the Boston Medical and Surgical Journal. It cost $3. It was delivered 4 times a year on horseback. Then in 1823, another guy in Boston, John Smith, starts a weekly medical journal. He called it the Boston Medical Intelligencer. And it got to be very successful. So in 1828, John Collins Warren buys it for $600. And they call it the Boston Medical and Surgical Journal, and they start publishing it that way in 1828. In 1921, the Massachusetts Medical Society buys the Boston Medical Surgical Journal for $1. Now remember, John Collins Warren had paid $600 for the Medical Intelligencer in 1828. I've got investments just like that, right? You pay $600 for it, and a century later it's worth a 600th of what you paid. And the dollars are worth less too. But they kept the name Boston Medical and Surgical Journal for 100 years, and then they changed it. But they kept the volume numbers in sequence. So the New England Journal of Medicine published in February, February 23rd, 1928, was a volume number that went back to 1812. So that allowed us to claim to be the longest continuously published medical journal in the world. We've never missed an issue, and we're not going to miss it on my time if I can do anything about it. But this is patients with tuberculosis. This guy is advocating. Everybody thought fresh air was important. First of all, they're fresh air. They were out of town, so they're probably away from you. But the big issue was, do you take people and put them at rest, or do you put them at exercise? And there were kind of 2 camps, and no one really knew what to do. It turned out the rest camp was winning. It's easier to have a bunch of people in a bed than it is to have them out doing push-ups and sit-ups and riding horses and stuff. Appreciated a great benefit came to consumptive cases, that's the tuberculosis, by a generous diet and outdoor life, horseback camp, another outdoor to be quite recommended by the profession, right? But it turns out that he comes out at the end of this saying you're better off in bed. So it took, let's see if I can get this thing to go forward, this guy, so this is a guy named Burns Amberson. And he was born in Ohio, but he went to Johns Hopkins Medical School. And he graduated in 19 19. Pretty sure it was 1919. And he not only graduated with a degree, he graduated with tuberculosis, which he didn't have when he came. And that was very common among medical students. So instead of going off to do an internship, he went to a tuberculosis sanitarium. Now, he was fairly well off, so he went to the Loomis Sanitarium, which is in Liberty, New York. Those of you familiar with it, it's sort of part of what we now call the Borscht Belt. But this is an upscale sanitarium. This shows it. It shows the administration building. And he was there as a patient, and then because he was a trained physician, he worked on the staff. And he married there, and he had kids there, and he was being treated for tuberculosis. And finally, after 9 years, they say, okay, you're cured, out of here. So while he was there, and now I have to go on speculation because his records are not fully available. So this is information that I've intuited from what is on the written record. He was treated with sancroicin. Now, sancroicin, as you'll hear, is a sodium gold thiosulfate, which was a treatment developed in Denmark and shown in vitro to kill the tuberculous bacillus. And it was given to patients. And it is highly likely that Burns Amberson received this treatment, and he was convinced that the treatment almost killed him rather than helped cure him. You know, but that's the way it was. You take the cure, it's painful, but then you're cured. So here he is. He's out of this job. What can he do, right? He spent the 9 years after medical school in a TB sanitarium. So he gets a job in another TB sanitarium. Right? And the one he went to was the Spring Hill Sanitarium, which is outside of Detroit. And shortly after he shows up there, the guy who owned all the land around the Spring Hill Sanitarium was a real estate developer, and his wife had died of TB in that sanitarium. Now, he's a real estate developer. He realizes that the land around a TB sanitarium is worth zero. Like, nobody wants to be near a TB sanitarium. So what do? You give it away to the city, and that way you get your name over the gate. It's the Mayberry Sanitarium, and it's a memorial to his wife who died. And here it is. This is a children's ward. You see they're in rest. Do you imagine what it must take to keep 5-year-olds in bed like that? Right? I don't know if any of you have kids, but it's impossible. The windows are open. This is what life in a TB sanitarium was like. And this, as you can see, is it's not first-class accommodations. It's not like where Hans Kastorp was in Zauberberg, the Magic Mountain. And here we see him. Staff picture. That's why you should always take a staff picture. You might be famous one day and people will haul it out. So what his idea is that this stuff is bad for you. So he publishes this study. It's a clinical trial of sancroicin. And he says in 1924, remember he was in the sanitarium in New York from 1919 to 1928. Molgard published results of the study of sancroicin and made claims the substance had specific curative effects on tuberculosis. All in vitro studies, no studies in humans, but then studies were done in humans and they said, yes, it looks like it works. So Burns Amberson does something that nobody else has done. He and one other guy on the staff, and I don't know who is who. I only know Amberson from his other pictures. What he did was he made 2 groups of patients that were exactly the same, as far as he could tell. 12 patients in each group. They had the same temperature, they had the same x-rays, they were the same age. He tried to match them as good as he could. But then he did something that nobody else had done before. He randomized them. By the flip of a coin, one group became the treatment group, and one group became the control group. And this is a huge step forward because when people had done trials before that, the docs were assigning patients. You know, Kay, you look okay, or Taddy, you look okay. I'm going to put you in the control group. You look bad. I'm going to put you in the treatment group, or the other way around. You know, it was subconsciously. They weren't thinking about this. They were just kind of doing it. They weren't letting the play of chance, again, decide. But Amberson started randomization, created 2 groups that were equal, which he could do because it's a single-center study, and then he and the other guy working with him, knew which group was which, but nobody else did. Kept it a secret. So he blinded at the same time. And here are his outcomes. This is a This is tables. This is tuberculosis grading over here, and this is treatment. So this is active treatment. This is control. I got nothing. So this is slightly improved versus much worse. One died versus treatment. I probably would have put that in the much worse category myself. Missing data. Patient died. But you can see that of the patients with tuberculosis, 5 were slightly improved, 4 were much worse, 1 died during treatment. Patients who got the control treatment, 6 were slightly improved, none were much worse. And he concludes this stuff doesn't work. And people stop using it. Now, it took a while, that was mid-1930s, for this man, a Hungarian microbiologist working at Rutgers, Selman Waxman, working with fungus. He was interested, if you have more spare time after you've finished Der Zauberberg, The Magic Mountain, you can read My Life with Microbes by Selman Waxman. And I was interested in soil, bacteria, and fungi. Not many people were interested in funguses, so he pretty much had the field to himself. But an isolation from Streptomyces, a fungus, was streptomycin, and he could make small amounts of this stuff, and it would actually cause the tuberculous bacillus to die. And then he partnered with some people at Mayo Clinic and showed that guinea pigs who were given tuberculosis could be cured with this streptomycin. But then he had a quandary. He couldn't make enough of this stuff to test it in humans. So what did he have to do? He partnered with a drug company. Okay? This is important. He realized that he wasn't going to get from the bench to the bedside without help. And he partnered with Merck. And the people at Merck developed a process to grow enough of this stuff and extract it so you could get made adequate amounts under what we would now call GMP, good manufacturing practices. It's probably different back then, but stuff that you would give to a patient without worrying there was a lot of crap in there that was going to kill him, that wasn't the drug. The drug might kill him, but that was on purpose. And so he partnered with a drug company. And I think that one of the things that's happened to our medical industrial complex is that we're building a wall between academics and drug companies. And I think that we need to tear the wall down. We've got to work with them. We have different goals. We can't be bought out by them. But we have to work with them. And I think we've seen it again with Ebola. GSK is making vaccine. There's no university that can turn out 10 million vials of vaccine. We have to work with the drug company. And they have a big investment in stuff like this. So he works with the drug company, and an aliquot of that goes to England to be studied in the British Medical Association, and streptomycin treatment of pulmonary tuberculosis. This is another kind of interesting aside about journals. If you had big medical information to publish, of course this is an English study, in the 1930s, the 1940s, the 1950s, you published it in the BMJ. That was the place to publish stuff. And what they do here is they describe their study of pulmonary tuberculosis with streptomycin. Double-blind, placebo-controlled. Now, they had problems that Burns-Amberson didn't have, because they were recruiting patients at many centers throughout England. So they needed a statistician, Bradford Hill, who was spending his time inventing the statistics as the study was being done. And that's what we had to do. And I think that as we confront new problems in genetics and in personalized medicine, we have to invent new ways of analyzing what we do. It's perfectly okay. You just have to be honest about your inventions and show that they have value. And so they're able to study patients because he came up with a way of randomizing them. And what they did was they had a way of randomizing patients at many sites. Here are their data. So this is now stratified by temperature. Remember I told you from Hans Kastorp's thermometer story. Low temperature, middle temperature, high temperature. Notice they hadn't converted to Celsius at this point. And if you're above the line, you're improved. If you're below the line, you're worse. If you're black, you're dead. This is streptomycin. This is control. This is the key up here. And you can see by inspection that the S group did better than the C group. There's not a p-value in this entire article. He says in the text that the likelihood of these differences occurring by chance alone was less than 1 in 100. But there's no p less than so on. We hadn't gotten around to using p-values that much in the early 1940s. So that with this, the randomized controlled trial comes of age. So this is an n-gram, right? Everybody know what an n-gram is? If you're under 20, you'll know what it is. If you're under 30, you might. And Teddy, you probably don't know what an n-gram is. So you go to Google, and you type in a word or a phrase, and you say to it, how often does this word, phrase, occur in the corpus of digitized information that Google owns? And then, a few milliseconds later, you get an answer. And if you're looking at the words clinical study, it started appearing in 1840. Now, some of this is biased by the amount of literature they've got, but there's enough of it here, and then it starts taking off in the 1940s, goes up. But randomized controlled trial, clinical trial, or controlled trial, either one, doesn't really start showing up in big numbers over here as the percentage of digitized material until the mid-60s. So this is relatively recent. This is a change in the last 50 years. But we were there at the beginning. We saw Burns-Anderson introducing randomization and blinding, the big TB trials. I didn't tell you that the second article, the first article in the British Medical Journal is the stuff works. Second article is a number of patients developed resistance, back to back, giving us another important thing. When you're working with a drug company, you want to tell the good news and the bad news, right? We don't want to have selective. Tell the whole story, then people know the whole story. It's not terribly complicated. Then we have all these drugs for tuberculosis. We have gold therapy introduced and gold therapy abandoned. We saw why it was abandoned from the work of Amberson. Then we have streptomycins introduced in 1943. We have all these drugs that have been introduced as a result of randomized controlled trials. So let's look at a new one now. Of course, it's from the New England Journal of Medicine. There are other people who publish them, but, I've got a bias here, which you know. So this is Medeliquin. This is actually an interesting drug. So they took patients with multi-drug resistant tuberculosis. So these are people who are likely to die from their disease if you don't do anything. They're in this, some people would say, some bad shape. And they had 24 weeks of treatment in an investigational treatment phase, and then there was, after the drug was stopped, they went in to see if the drug is cured, the disease doesn't come back. It's you don't need Spinoza for that. So they were on Medeliquin plus their background regimen versus the background regimen alone. Then, so this is a 24-week evaluation, 120-week analysis. And then the other part of the group got placebo plus the background regimen. So they're not being hurt because they're getting the best treatment that they could get. So it's best available treatment plus new stuff. And the new stuff could hurt you. And as you'll see, it does. It also helps you. So this is the time to culture conversion. When you have it, what you would like for it to happen is everybody to have a 0%. A positive culture would be good. And you can see Medeliquin plus background regimen gets to lower culture rates than the background regimen. So this is good news. And when you look at this treatment outcomes, we've got death, withdrawal, treatment failure, cure. Now we have the conundrum. We've got, in the Medeliquin group, we have 58 patients are cured, 58% are cured, versus 32 with placebo. That's good, right? It's almost twice as many. You look at the deaths. There were 3 in this group, 12 in that group. Here at the FDA, do you approve this drug? You have twice as many people cured, 4 times as many people dead. Or you have 9 people dead for every additional 26 people you cure. It's a devil's bargain, right? It's not easy. The FDA approved this drug because the information's out there. So you're not trying to hide anything about this. But if you have multi-drug resistant TB, you're not only going to die, you're a danger to your community. And so the world's better off with a drug like this, even though there's probably a higher risk of death during the treatment period. There's a substantial risk of cure. So you can see how even in this is stuff that we've published in the last year, even now, in the year 2015, with this disease that's been around for centuries, we've got conundra that are not easy to deal with. But the thing that's important is that when you report a trial, you tell people the good news and the bad news, and then they can make a decision. We're not doing a marketing spin here. We're telling you the story. You and your doctor can work out what works for you. So, randomized controlled trials, compared to what we were doing before, is that they have improved power over observational data. I know as an epidemiologist you hate to hear that, but it's the way it is. But we needed to safeguard patients in trials, and we need to have public accountability. So I can do two things. I can stop now, or I can go on and tell you a couple of more stories. Who would prefer to do the extra stories? Who would prefer to ask questions? Got to finish by 10 o'clock. All right, so now I'm going to force you. Raise your hand if you would like to have me continue with this story. All right, there aren't enough people who didn't raise their hand. So, data safety monitoring boards. This is now part of the clinical trial scene. It started with the Coronary Drug Project. They were doing this big study looking at niacin as a way to lower cholesterol. And they were doing this study, and they realized, hey, wait a minute, we need to understand this better. And so the NIH, who was sponsoring the study, got Greenberg, who was a cardiologist, to give them an opinion, and he published the so-called Greenberg report. And they got non-participant experts, that is, people who are not part of the trial. And they're going to look at the trial, and they're going to say the trial is ethical to continue unless the question's been answered by the data in the trial. The data are so compelling that it doesn't make sense to continue to the planned end. 1. 2, that there was excess risk or benefit. Like the data might not have been compelling, but a lot of people were dying in the active treatment group. Or the landscape's changed. We have a trial of niacin here, and let's say this is ongoing and statins are introduced. You have a better therapy. It doesn't make sense to continue the trial. So that's what the DSMB does. And this is the clofibrate and niacin in coronary artery disease, was published in the JAMA. The NEJM had not reached its ascendancy. We really didn't reach our ascendancy until the 1980s. So this is where you published the big impact research in the 70s in the JAMA. And for those of you who are giving you a lot of reading recommendations here, David Demetz, Kurt Furberg, and Larry Friedman have this book on data monitoring in clinical trials. It's vignettes from 33 cases of data safety monitoring boards. And it's about as close to a page-turner as you can get in this business. They're really interesting stories of what it was like to monitor a clinical trial. And so this is one of those stories, because it follows from the one that we just talked about. It turns out that if you took clofibrate and niacin, lowered your cholesterol, you improved the likelihood that you were going to die. So here's an article published in the New England Journal of Medicine about ventricular premature beats, known as VPBs, and death after myocardial infarction. And here are the data from that. It's the cumulative probability of death. So you prefer to have a low score. It's a game like golf. And you found that if you looked at all causes of death and a patient had, at a point in time in enrollment, complex VPBs versus no VPBs, they had a higher risk of all-cause mortality. Now, they're in your office. They've got these abnormalities in their EKG. There's an association, right? So a way to think about this is there's causality here. If I could do something to get rid of those VPBs, I could probably prevent death, right? It seems not unreasonable to make that kind of assumption. So the drug companies went out and found a bunch of things that suppressed VPBs. And here we are, econide, flecainide, imipramine, and morcerazine. And this is a pilot study where they were able to show that each of these drugs, except for morcerazine, I think, prevented VPBs. So now they had drugs that could prevent VPBs, and we knew that VPBs were associated with death. So we want to do a study where we're going to take patients that have VPBs, and we're going to put some of them on drugs that prevent VPBs, and we're going to put other people on placebo, and we're going to see what happens. Right? There were people who said it's unethical to do this study. We know the outcome. We know those VPBs are bad. Right? We know we have a way to prevent them. We are putting these people at risk. It's like jumping out of an airplane without a parachute. You're a bunch of pointy-headed academics. get your act together, you're fools. But they did it anyhow. The NIH sponsored the study. You had a heart attack within the past couple of years or past week, and then they had a run-in period. This is personalized medicine. You were tried on one of 3 drugs to see which one of them would suppress your VPBs. If you couldn't find one that suppressed your VPB, you're out of the study. You found one that suppressed your VPB, you were in the study. So now you're in that study, your VPBs are suppressed, and you're randomized to receive the antiarrhythmic drug or the placebo. Now at this point in the history of clinical trials, the Data Safety Monitoring Board was only looking at the data as Group A versus Group B. So here they are, Group A versus Group B. You're watching a trial. You're on the Data Safety Monitoring Board. It's been going on for, almost 2 years. You're looking at survival between the 2 groups. You say, look, it's working, right? Those people in Group A are doing a lot better, right? This is great. And so finally they decided the difference was so big that they maybe ought to do something about protecting those poor people in the placebo group. So they unmasked. And this is what they discovered. The placebo group was doing better than the active treatment groups. Right? It was exactly what people didn't think would happen. Right? If they hadn't done the trial, we'd still be giving these drugs. People would be dropping like flies. Not like flies. You can see it's not all that big a difference. You'd never see that. 6 cases per 100. But they studied it. They didn't go through the reasoning and saying the reasoning was so compelling we shouldn't do a trial. I have surgeons coming to me, it's usually surgeons coming to me all the time saying, we can't do a trial, we know the answer. And I always say, if it's so clear, your effect size will be really small. You'll be able to do this in a week. Prove it. I'm from Missouri. You have to prove it. That's your job. You're here to show that something actually works. Something that we think we know becomes something that we really know. That's what the game is about. It's published in the New York Times. This is interesting aside on medical publishing. This is Wednesday, April 24th, 1989. Warning issued on 2 heart drugs. New York Times, front page above the fold. My predecessor, 4 times removed, Bud Relman, rushed this to publication. This data came available in April of 1989, and it showed up in the New England Journal of Medicine in August of 1989. Right? April to August. That was the super rush in 1989. We've gotten articles in now. We have articles on Ebola. We have articles in on tainted heparin, in and out of the office, peer-reviewed, revised, and up within a week or less. Because when the public health's at stake, people should be able to see the data. The scientific information shouldn't just read about it in the New York Times. So I want to thank a couple of interesting people. This is sort of like Mr. Tall and Mr. Short. Will Carspeckin, the guy on my side wearing the bow tie, is an anesthesiologist on the West Coast. He's in the 2nd year of anesthesiology training. And Ming Zhai is also on the West Coast studying to be a radiation therapist. They helped a lot with these images. And then on your right-hand side, we have Celestine Warren, who is the great-great-great-great-great-great-granddaughter of our founder, John Collins Warren, and a student at Harvard College. Thanks for your attention. I'll be happy to answer questions. So Kay says there's time for 1 or 2 questions. Speak now or forever hold your peace.
Students can meet afterwards for 1 hour with Dr. Drazen.
How come somebody has to have a question? How come you have more snow in Baltimore than we do in Boston? There's an answer for that. What's the answer? No? Right, the high pressure was, in Boston, it was like really wicked cold, and that high pressure came down, and the boundary between the high cold air and the warm air from the south, instead of hanging out in Boston, was hanging out over Baltimore. My son sent me a picture. He lives in Potomac, digging out. This kid's had the day off of school. He had the day off of work. Yes?
Yeah, enjoyed your talk. All of us in this room probably put clinical trials at the top of the evidence pyramid, and yet they were the first, I think, big casualty with the funding crisis at NIH. So, what are your thoughts here? Because there's a total schism between, I think, what journal editors, what professional societies want, and the funding mechanisms that now are available to people who want to do trials.
So there are kind of 2 answers to that question. One is that people at the NIH have to decide what they can support, and they are supporting some clinical trials. They haven't given up entirely. They've changed their agenda. The cynics, the Tea Party types, say that drug companies develop drugs, they do clinical trials. Why should the NIH get in this business? Now, when a drug's brand new and a drug company doesn't want to give away rights to it, I can see why they do the trials. I think that for the sake of the public health, it makes sense to study drugs by dispassionate third parties. But it has to at least get to the point where the drug's on the market so somebody could buy them to study them. So it's a very complex calculus. I'm not Francis Collins. I'm not quite sure. We need to have a balance between basic science research, where we discover new things that could lead to even more effective therapies and diagnostics, and we have to have some clinical research. And I'm not quite sure if our current study section methodology is the best way to make that decision. And one of the things that he's tried to do, Francis has tried to do, by appointing clinical directors to the various institutes, is to give them that priority to be able to decide what needs to be studied and what doesn't need to be studied. And there's always going to be people who disagree with them, because it's like any other human endeavor. If everybody agrees, it's boring. So my personal opinion is I would like to see more clinical research, but I'm not the guy calling the shots, and I can see that if we only did clinical research, we wouldn't get the basic information we need. So it's a very tough call, and I don't think it's an easy way to make that separation. Yes?
Yeah, if you could just switch back to that slide where you showed the conundrum between people, with more tests with the treatment and less tests without treatment. The one you were just pause.
I'm trying to see if I have to turn this thing back on here. I turned it off to save electricity.
Yeah,
I know. This one. Yes.
So my question was that since these people with a placebo, there's more treatment failure. What does that mean? I mean, wouldn't those people already be dead either way?
Well, so they're on background therapy, but what you do when you treat tuberculosis is even though the treatment's failed, you continue on, because if you stop the background therapy, the disease accelerates. So they're not reaching cure. What you're thinking is that the side effects are worth what you get. Then there are people who are withdrawn because their regimens, and anybody that's tried to treat patients with multi-drug resistant TB, the drugs are terrible. I mean, it's no fun to be a multi-drug resistant TB patient, and patients can't stand it. Doctors don't want them. They're having so many side effects from therapy that and they end up dying, but they haven't died so quickly. You don't die instantly from tuberculosis. You die over the course of a long time, and then you have a problem with somebody with MDR TB that you have to keep isolated. You don't want this guy run down on your community. You don't want to be sitting next to him on the airplane and having him or her cough the whole time. So, yes?
One was also the size of the groups. And 3% of the statistics seems like there was like one death versus 3 or 4 deaths. I wonder if the number of people that die, the percentage of people that die is very small to be precisely estimated, that it could be that we have similar death rates, but very few.
So you're saying a group of 66 people is not enough to have an accurate estimate of the true death rate. That's a possibility. It could be the play of chance. When they worked out the numbers here using statistics, they're able to show there's a significant increase in death. 3 versus 12 worked out to be significant. It could in part be the play of chance. Okay, I think it's time to have a break, and for the students that want to hang out. For those of you who want to leave, please.